next topic we are going to deal with is morphology of bacteria. Since we have seen different microbes, different branches of microbiology, the first branch we are going to see is bacteriology. So, like you learned anatomy and physiology, human anatomy and human physiology in your first year classes, when you learn about the microorganisms, you should know about the microbial anatomy and microbial physiology also. So for bacteria, we'll start with the bacterial anatomy. So when you won't say bacterial anatomy, you can modify the term as morphology. So morphology of bacteria or the structure and anatomy of bacteria. So the first thing you should know about any kind of morphology is the size. So you should have a general idea about how big is the microbes or what is the size of the microbes. Coming to bacteria, the size is around 0.2 to 1.5 micrometer diameters and around 3 to 4 micrometers in length. So for ease of remembering, you can just learn it like 1 to 2 micrometers. So that would be good enough. 1 to 2 micrometers is the size of a normal bacteria. Most of the medically important bacteria comes within this size. Now, how you classify bacteria is based on the shape. So different bacteria have different shapes. Based on the shapes, we can classify bacteria broadly into two groups. That is cocci and bacilli. So cocci, when you say itself cocci, it has got an O. So the shape of cocci is like a O. It's spherical. And bacilli, remember there are two L in bacilli, no? So it is like L or rod shaped. So cocci is spherical and bacilli is rod shaped. Now we have very important, medically important bacteria coming under cocci. So though I told you it is spherical cells, it can also be oval cells, oval or spherical cells. Now, based on how they are arranged, you put them into different kinds. Like cocci, if it is in pairs, if a single cocci, some bacteria it will be seen as single cocci, but some it will be seen as pairs. Two cocci will remain together. That is called as diplococci. Diplo means two. So, diplococci example is pneumococci or streptococcus pneumoniae, which causes pneumonia. The name itself indicates pneumo means it is related to respiratory system or lungs. Now, some cocci, they appear as chains. You can see here, cocci appearing as chains like this. So, it is called streptococci. The term strepto means chains. So, streptococci is the bacteria which is seen as chains of cocci. Some it will look like a bunch of grapes. You can see here cocci in groups. So that is staphylococci. Staphylo means bunch. So a bunch of cocci appearing together is called as staphylococci. That is example for cocci in clusters. And some cocci they will be looking like tetrads. 4 cocci will remain together. Example is micrococci. And I'll show you how these cocci look like diplococci, streptococci and all. That is, if it's a coccus, when a coccus divides like this, bacteria divide by binary fission. So when it divides by forming like this, two cocci, two daughter cells are formed and they're very fond of each other. They don't separate. They don't get separated from each other. They remain as diplococcus. So there is division in one plane and they remain together. That is diplococcus. Sometimes what happens, division is in one plane itself and none of these daughter cells are separating. They form a chain, that is streptococcus. So chain of cocci is seen in streptococcus. And I hope you remember diplococci example is pneumococci. Pneumococci is the example of diplococci. And when the division takes place in two planes and they fail to separate, they will form a tetrad. And that tetrad example is micrococci. So what happens is in a cocci, if division occurs in one plane, see, the division occurs in one plane and they fail to separate, they're very fond of each other, they don't separate, the daughter cells remain together, then it forms diplococci. So what is the example for diplococci? Pneumococci. Pneumococci is the example of diplococci. Okay, then what happens is, suppose the daughter cells are dividing continuously and none of them get separated from each other, they form a chain, then it is a streptococcus. So, diplococci example is pneumococci, bacilli in chains example is streptococcus. And sometimes the division occurs in two planes. You can see two planes and four daughter nuclei daughter cells are formed and four of them remain together. They don't separate from each other. Then they form tetrads. So tetrad example is micrococci. And when division occurs in multiple planes, you can see here multiple planes division occurs and they are failing to separate. They form a cluster or bunch. An example for bunch of cocci is staphylococci. So it is because of the division in various planes and the daughter cells failing to separate from each other, they lead to different kind of arrangements. So that is about cocci. Now coming to the next type, I told you bacilli. Bacilli has got two L, so they are rod-shaped organisms. They are elongated rod-shaped organisms. So bacilli, normally a simple bacilli, when it looks like this in staining, it will be in different alignment. The alignment may be different, but they look like this simple rod-shaped bacilli. The most important example for bacilli, Bacillus anthracis, the name itself is bacillus. So it is bacilli. E. coli, Klebsiella, Salmonella, those bacteria are bacilli. Sometimes what happens is this bacilli, they will be having equal width and equal length. Like they will look like a cocci. They are bacilli, but they have width equal to length. So when you see under microscope by staining, they look like cocci. Some will look like bacilli. So then you call it as Coccobacillus. So example for a Coccobacilli is Brucella. Actually it is a bacilli, but because of the equal width and length, they look like cocci. Some bacilli, like streptococci, you have seen chains of cocci. Some bacilli also fail to separate from each other after division. They form long chains of bacilli. Example is streptobacilli. And the last one is cuneiform arrangement. You can see here palisading, that is some bacilli, they will form angles with each other. They will look like V or L, letters, alphabets V or L. So that is called cuneiform arrangement. You also call it as Chinese letter pattern. So which bacteria is seen in this pattern? It is corine bacteria. Corine bacterium, they form angles with each other. They look like V or L-shaped letters. That is called as Chinese letter pattern or cuneiform arrangement. So these are some of the arrangements of bacilli. Now coming to some different shapes of bacilli. The one which causes cholera, Vibrio, the name cholera. Remember, cholera is comma-shaped bacilli. Cholera is comma shaped. You can see here comma shaped bacilli. It's slightly curved, so it looks like a comma. And some other bacilli like spirillum. Spirillum, the name itself indicates it's spiral shaped. And they are very rigid spirals. You can see here they are spirals but very rigid bacilli. So spiral shaped bacilli example is spirillum. So these are some of the alterations in the shapes of bacilli. Not like regular rod shape, they get slightly curved or they become spiral. You have different shapes to different bacteria. Now the different ones from all these. So there are another group of bacteria which has got a different kind of shape. Like the spirilla which we mentioned just before, spirochetes is another group of bacteria under which comes the causative agent of syphilis, Treponema pallidum. You can see how they look like. They are also spiral like spirillum, but they're very thin spirals. So very thin spirals are seen as the bacteria which look like very thin spirals are spirochetes, and the important examples of spirochetes are Treponema pallidum, then the one which causes Weil's disease, leptospirosis, leptospira. All these are very thin spirals. You cannot visualize them under light microscope. They are very thin. Now another group of bacteria here, you can see lot of bacilli in branching filamentous form looking like fungus. So that is the group called actinomycetes. So actinomycetes are the branching filamentous bacteria. Here you can see multiple bacilli in multiple branching filament like appearance, like this they will look like. Now apart from all these, so we have seen cocci, examples of cocci, bacilli, examples of bacilli, and the alterations in the shapes of bacilli like curved or spiral shaped or some looking like Coccobacilli, Chinese letter pattern. All these, apart from all these, there are two other groups of bacteria which are more or less similar to viruses. Rickettsia and Chlamydia. Why they are like viruses? Because they are very small and they are obligate intracellular parasites. They can live only inside cells. So that is a property of viruses. So these bacteria, they have some similarity to viruses, rickettsia and chlamydia. And the other one, mycoplasma. Mycoplasma, the importance is it doesn't have a cell wall. It doesn't have a cell wall, so it does not have a shape, no shape. If there is no cell wall, the bacteria won't be having any shape. So mycoplasma is the cell wall deficient bacteria. Rickettsia and chlamydia are like viruses because they are obligate intracellular parasites. And when you hear about actinomycetes, the term comes like mycology, mycotic infections, closer to fungus. Why they are called like that? Because they are seen as branching filaments like fungi. And also remember spirochetes, very thin spiral bacteria, example is Treponema and Leptospira. So with that, we finished the classification of bacteria based on the shapes. Now we are going to see the bacterial structure, internal structures, each part of the bacterial structure in a little bit detail, because each of the structures can be asked as questions in the university papers. So at the end of the class, we'll see what are the previously asked questions from this portion. So here you can see the bacterial structure, what all things you can see. Outside, there is a long thing which is protruding from this bacilli, this red thing, a very long thing. That is flagella. What is the use of flagella? With the help of flagella, this bacteria can move. So flagella gives motility to the bacteria. It acts as a propeller function. Now there are very small, thin, blue color things protruding from the cell. What may be them? That is fimbria or pili. They are helpful for attachment of bacteria to the host cell surface. Bacteria needs something to get attached to the host cell surface. Only then it can invade and cause diseases. So for attachment, this pili helps. Then comes this yellow color, this black color thing which is surrounding the yellow color layer. That is capsule. It is not present in all the bacteria, only some bacteria will have capsule. We'll see what it is. And beneath capsule, there is that yellow color thing is the cell wall, like the other cells which we have learned before. This is also having a cell wall. Bacteria has a cell wall, and beneath cell wall, there is a cytoplasmic membrane, like you have learned the human cells or plant cells in your school classes. Here also there is cell wall, cytoplasmic membrane, and cytoplasm. And inside you can see there is no properly formed nucleus. Normally in a cell you will see a properly formed nucleus with the nucleolus. Here you can't see a nucleus, you can't see a nucleolus. It is just having a DNA. It's just a nuclear material without a proper nuclear membrane. So that is why bacteria is put under prokaryotes. Right. And you are not able to see the normal cell organelles we see in other cells, like there is no endoplasmic reticulum, there is no mitochondria, and there is no Golgi complex. Nothing is there. And this is having only ribosomes. See, bacteria has got ribosomes for protein synthesis. And instead of mitochondria, they have some invaginations from the cell membrane called as mesosomes. So if this is a bacterial cell membrane, there are some invaginations of the bacterial cell membrane that is called as mesosomes, which is in addition, which is in plays the role of mitochondria because it doesn't have a mitochondria. So they don't have the normal cell organelles which we see in the eukaryotic cells, but it is having ribosomes and mesosomes. And in addition to the chromosomal DNA, you can see circular extrachromosomal DNA which is called as plasmid. So these are the layers or structures of bacteria. So we'll see each one by one, only ones which are important for us in exams. First one is cell wall. I tell you, cell wall is a question for exam. It can be asked as a question in exam. And how you will learn cell wall is different in different type of bacteria. So after this, you will see the staining technique. So then you are going to learn the most important staining technique we use in microbiology is gram staining. So this gram staining helps in distinguishing or differentiating bacteria into gram positive and gram negative. Why this happens is gram positive bacteria has got a particular type of cell wall and gram negative bacteria has got a different type of cell wall. And in this picture, it is very clear, and I have given a mnemonic also for that. We'll see what it is. See, in gram-positive cell wall, what happened? There is a thick blue colored layer, no? That blue colored layer is called as peptidoglycan. And what happens to this peptidoglycan in gram-negative bacteria? It's very thin, right? So that is one major difference between gram-positive and gram-negative cell wall. Peptidoglycan layer is very thick in gram-positive, it's thin in gram-negative. And you can see, in addition to peptidoglycan, in gram-positive, there is certain things coming from this cell wall. That layer is called as teichoic acid. So this is something which is present only in gram-positive cell wall. And in gram-negative, what you're seeing is peptidoglycan is very thin, but there is an outer membrane which is composed of lipids and polysaccharides and proteins. So lipids, polysaccharides and proteins. So these three are present in gram-negative cell wall. So you call that lipids plus polysaccharide as lipopolysaccharide. Lipopolysaccharide layer. So that is the thicker layer in gram-negative cell wall, lipopolysaccharide, and also you have outer membrane proteins which is called as OMP. So they don't have teichoic acid, their peptidoglycan layer is very thin. Instead, they have a very thick LPS layer, lipopolysaccharide layer, which constitutes the outer membrane. Also, they have the outer membrane proteins. And between, so this is the plasma membrane, which is present in both. Plasma membrane or cytoplasmic membrane is present in both. So between this plasma membrane and the outer membrane, gram-negative bacteria has got a space called as periplasmic space. Periplasmic space. So this periplasmic space is constituted by peptidoglycan layer. So in summary, you can remember gram-positive has got peptidoglycan and teichoic acid. Long PPT, that is a mnemonic. We hate long PPT, no? So PPT, positive peptidoglycan, teichoic acid. So gram-positive bacteria has got a thicker peptidoglycan layer and also a teichoic acid layer. Whereas gram-negative, long is lipopolysaccharide, outer membrane, negative, gram. So gram-negative bacteria has got lipopolysaccharide and also outer membrane protein. So that is the major difference between gram-positive and gram-negative cell wall. This is important in staining. That is why one group appears in a particular color and the other one appears in a particular color. Here you can see the positive color is violet and the negative color is pink. So gram-positive bacteria will appear as violet color and gram-negative bacteria will appear in pink color. So this is important for exam. What is the difference between gram-positive and gram-negative cell wall? So with the help of that image which you have seen just now, you can remember that. What is that? Thickness. So comparing the thickness of both the cell walls, which one is more thicker? This peptidoglycan layer is a cross-linked layer. It has got multiple residues which is a cross-linked. It will be like different layers will be there which will be cross-linked to each other. So it's a very thick layer, peptidoglycan layer. So thickness is more in gram-positive bacteria. Gram-negative is comparatively thinner. What about the peptidoglycan layer? It's 16 to 80 nanometer size in gram-positive. It's just 2 nanometer in gram-negative. And which one has got teichoic acid? It's present only in gram-positive. In gram-negative, it is absent. And lipopolysaccharide, it is absent in gram-positive. It is present in gram-negative. And periplasmic space is also present only in gram-negative. So with the help of a mnemonic, you can easily remember peptidoglycan and teichoic acid are present only in gram-positive, lipopolysaccharide layer, outer membrane layer, and periplasmic space, it is present only in gram-negative. So these are the differences between gram-positive and gram-negative cell wall. And what is the role of cell wall? Cell wall gives shape to the bacteria, it gives rigidity to the bacteria, and it acts as a site of action of most of the antibiotics. Most antibiotics act in the bacterial cell wall. That is how they inhibit the generation or replication of bacteria. Okay. Now, when we finish cell wall, you should know what is L forms. It may be asked as a 2-mark question. L forms. So, when you learned about cell wall, you should know another form called L form. L form, why it is important is, it may be asked as a 2-mark question. And what is L form is cell wall deficient form. Some bacteria, when they replicate, the cell wall synthesis may be inhibited. This happens especially in the presence of antibiotics like penicillin or enzymes like lysozyme. So when bacterial division is inhibited by certain antibiotics or lysozymes, it is the cell wall which is affected. Cell wall synthesis is inhibited. In that case, bacteria will try to replicate without cell wall. So cell wall deficient forms may be formed, and that is called as L forms. So sometimes it can be asked as a 2-mark question for exam. And what happened in gram positive cell bacteria when this happens is they form protoplast. That is, this cell wall synthesis is inhibited, I told you, and most of the case, the layer which is affected is peptidoglycan layer. So we know peptidoglycan layer is present only in gram positive. The thicker layer is present in gram positive. So when this happens in gram positive bacteria, the peptidoglycan won't be formed, so they won't be having a cell wall at all. So protoplast is a form which doesn't have a cell wall. Whereas gram-negative bacteria, they will be having, even if it is an L form, it will be having some cell wall because they have the lipopolysaccharide and all. So that form is called as spiroplast. So protoplast in gram-positive and spiroplast in gram-negative. Both of these are L forms, which is due to inhibition of the cell wall synthesis. And why L forms are important is bacteria can form L forms and that can lead to chronic infections. So we give antibiotics, we will think that the bacteria will get inhibited. But what they do is they are even more smarter. They will replicate without forming cell wall. So cell wall deficient forms will be remaining there and they can cause chronic persistent infections. So that is what is called as L forms. So we have finished cell wall. Cell wall is important for exam. Gram positive, gram negative cell wall difference is important. So please study that very well. And with this, we conclude this class and we'll continue with the morphology of bacteria in the next video. Thank you. So with that, we finished the morphology of bacteria. We have seen cell wall, capsule, flagella, fimbria, spore, all are important questions. So then now we have to move on to staining and microscopy, the different staining methods which we'll take in the next video. So with this, I conclude. Thank you.